Plant kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker in 1969, wherein he suggested the five kingdom classification, that is Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with further classification with kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So the cyanobacteria, that are also referred to as blue-green algae, are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperm and angiosperm under Plantae. Let us also look at classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological character such as habit, color, number and shape of leaves, etc. They were based mainly on vegetative character or on the androsium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against these natural classification system developed which were based on natural affinities among the organism and consider not only the external feature but also internal feature like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. These assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulty in classification. This become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed in this way. Each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy, that uses the chemical constituent of the plant to resolve confusion, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing simple thalloid autotrophic and largely aquatic, both freshwater and marine organism. They occur in variety of other habitats, moist stones, soil and wood. Some of them also occur in association with fungi, that is lichen and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Eulothrix and Spirogyra. It is given in figure 3.1. A few of the marine forms such as scalps form massive plant body. The algae reproduce by vegetative asexual sexual method. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores. the most common being the Jew spore. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes in dissimilar in size as in species of Eudorina. Eudorina is termed as an isogamous fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as ogamous. Example: Volvox and Fucus. Figure 3.1. There is example given of algae: green algae, brown algae, and red algae. Example of green algae: Volvox and Eulothrix. Example of brown algae: Laminaria, Fucus, and Dictyota. Example of red algae: Porphyra and Polysiphonia. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producer of energy-rich compound, which form the basis of the food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water holding substance. Example: algin in brown alga and carrageen in red algae, which are used commercially. Agar, one of the commercial products obtained from Galidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in protein, is used as supplement even by space travel, travelers. The algae are divided into three main classes: Chlorophyce, Phaeophyce, and Rhodophyce. 3.11. Chlorophyce. The member of Chlorophyce are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage body called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction show considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or ogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Cara. It is given in figure 3.1a. 3.12. Phaeophyce. The member of Phaeophyce or brown algae are primarily found in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocorpus, to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoid and xanthophyll. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrate, which may be in the form of laminarian or mannitol. The vegetative cell have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contain, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a hold fast and has a stalk, the stipe, and the leaf like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous and oogamous. Unit of gametes may take place in water or within the Ogonium Ogoimus species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. That is given in figure 3.1b. 3.13 Rhodophysi. The members of Rhodophysi are commonly called red algae because of the predominance of the red pigment R phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer area. They occur in both well-lighted region close to the surface of water and also at great depth in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have a complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile non-motile gametes. Table 3.1 Division of algae and their main characteristics. Classes, that is chlorophysi, phaeophysi, and rhodophysi. In chlorophysi is the common name of chlorophysi is green algae, and the major pigment present is A and B. Stored food is starch, and the cell wall is made up of cellulose. Flagella and number of position of insertion is 2 to 8 equal and apical. Habitat is freshwater, brackish water, salt water. Phaeophysi, common name is brown algae. Major pigment present is chlorophyll A, C, and fucoxanthin. Stored food is mannitol and laminarian. Cell wall is made up of cellulose and algin, and the flagella and position of insertion is two unequal and lateral. Habitat is freshwater, rare, brackish water, salt water. Rhodophysi, common name is red algae. Major pigment present is chlorophyll A and D. Phycoerythrin. Stored food is Floridian starch. Cell wall is made up of cellulose, pectin, and polysulfate esters. Flagellar number and position of insertion is absent. Habitat is freshwater, some brackish water, salt water, and salt water में most रहते हैं. Gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilizer development. The common members are Polysiphonia, Porphyria, Glaciria, and Geladium. 3.2 Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded area in the hills. Figure 3.2. Bryophytes, a liverwort. Merchantia A. Female thallus and B. Male thallus. Mosses. C. Funaria gametophyte and Sporophyte. D. Sphagnum gametophyte. Bryophytes are also called amphibian of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rock soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect, and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structure. The main plant body of the bryophyte is haploid. It produces gametes, hence it is called a gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is antheridium. They produce biflagellate antherozoids. The female sex organ called archegonium is flask-shaped and produce a single egg. The antherozoids are released into water where they come in contact with archegonium, and antherozoids fuses with egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animal species of sphagnum. A moss provide peat that have long been used as fuel and as packing material for transshipment of living material. Because of their capacity to hold water, mosses along with lichens are the first organism to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.21 Liverworts. The liverworts grow usually in moist shady habitats such as bank of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thylakoid. Example Merchantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy member have teeny leafy-like appendages in two rows on the stem-like structure. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structure called gamete. Singular gamma. Gamete are green multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamete become detached from the parent body and germinate to form new individual during sexual reproduction. Male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into foot, seta, and capsule after meiosis. Spores are produced within the capsule. These spores are germinate to form free living gametophyte. 3.22 Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched, and frequently filamentous stage. The second stage is leafy stage, which developed from the secondary protonema as a lateral bud. They consist of upright slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. These in this stage bear the sex organ. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of the leafy shoot. After fertilization, the zygote develops into a sporophyte consisting of foot, seta, and capsule. The sporophyte in mosses is more elaborate than in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria polytrichum sphagnum, figure 3.2. 3.3 Pteridophyte. The Pteridophyte include horsetail and ferns. Pteridophyte are used for medicinal purposes and as soil binders. They are also frequently grown as ornamental. Evolutionary, they are the first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about these tissue in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil condition. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in Teridophyte, the main plant body is Sporophyte, which is differentiated into true root, stem, and leaf. Figure 3.3. These organ possess well differentiated vascular tissue. The leaves in Pteridophyta are small, that is called microphyll as in Selaginella, or large, that is called macrophyll as in ferns. The sporophytes bear sporangia that are subtended by a leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones. Selaginella equisetum, example of cones or strobili. The sporangia produce spores by meiosis in spore mother cell. The spore germinate to give rise to inconspicuous, small but multicellular. Figure 3.3. Example of Pteridophyte: Selaginella equisetum and fern and Salvinia. Free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, and sandy places to grow. Because of these specific restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophytes bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of antherozoids. The male gametes released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophyte. In majority of the pteridophyte, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produces two kinds of spores, macro, that is large, and micro, that is small spores. Small spores are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygote into young embryo takes place within the female gametophyte. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, example Psilotum; Lycopsida, example Selaginella, Lycopodium; Sphenopsida, example Equisetum; and Pteropsida, example Rhiopteris, Pteris, and Adiantum. 3.4 Gymnosperm. The gymnosperm, gymnos means naked and sperma means seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after the fertilization. The seed that develop post fertilization are not covered, that is, are naked. Gymnosperm include medium sized trees or tall trees and shrubs. Figure 3.4. One of the gymnosperm, the giant redwood tree sequoia is one of the tallest trees species. The roots are generally tap roots. Root in some genera have fungal association in the form of mycorrhiza pinus, while in some other cycas, small specialized roots called coralloid roots are associated with antofixing cyanobacteria. The stems are unbranched, cycas or branched. Pinus, example Pinus and citrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extreme of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form legs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. These reduced gametophyte is called pollen grains. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same trees, that is pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cell of the nucellus. The nucellus is protected by envelope and the composite structure is called an ovule. The ovule are born on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form four megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears two or more archegonia or female sex organ. The multicellular female gamete is also retained with megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and the female gametophyte do not have an independent, free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air current and some in contact with the opening of the ovule, borne on megasporophylls. The pollen tube carrying the male gamete grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia following fertilization. Zygote develop into an embryo and the ovules into seed. These seeds are not covered. Figure 3.4 Gymnosperm example is Cycas pinus and Ginkgo. 3.5 Angiosperm. Unlike the gymnosperm where the ovules are nicked in the angiosperm or flowering plant, the pollen grains and ovules are developed in a specialized structure called flowers. In angiosperm, the seeds are enclosed in fruits. The angiosperm are an exceptionally large group of plants occurring in wide range of habitat. They range in size from the small wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, the Dicotyledons and the Monocotyledons, given in figure 3.5. The Dicotyledons are characterized by seeds having two cotyledons, reticulate venation in leaves, and tetramerous and pentamerous flowers, that is having 4 or 5 members in each flower whorls. The monocotyledons on the other hand are characterized by single cotyledon seeds, parallel venation in leaves, and trimerous flowers having 3 members in each floral whorls. The male sex organ in a flower is the stamen. Each stamen consists of slender filament with an anther at the tip. Within Within the anther, the pollen grain mother cell divides by meiosis to produce microspores which mature into pollen grains. The female sex organ in flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style, and stigma inside the ovary. Ovules are present generally. Each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspore. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has a three-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cell and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anther are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grain germinate on the stigma and the resulting pollen tube grow through the tissue of the stigma and style and reach the ovule. The pollen tube enter the embryo sac where 2 male gametes are discharged. One of the male gamete fuses with the egg cell that is called syngamete to form a zygote. The other male gamete fuses with a diploid secondary nucleus to produce the triploid primary endosperm nucleus, that is PEN. Because of the occurrence of two fusion, that is syngamy and the triple fusion, this event is termed as double fertilization, an event unique to angiosperm. The zygote develops into an embryo with 1 or 2 cotyledons, and the primary endosperm nucleus develops into endosperm which provide nourishment to the developing embryo. The synergids and antipodals regenerate after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. Figure 3.6, life cycle of an angiosperm. 3.6, plant life cycle and alternation of generation. In plant, both haploid and diploid cell can divide by mitosis. This ability leads to the formation of different plant bodies, that is haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte following fertilization. The zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by these plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gamete and spore producing during spore producing diploid sporophyte. However, different plant group as well as individual representing them differ in the following pattern. First, sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote result in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as Volvox, Spirogyra and some species of Calypnodomonas represent this pattern that is given in figure 3.7a. Point number 2. Point 2. On the other extreme is the type of wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic. And algae fucus represent this pattern. In addition, all speed-bearing plants, that is gymnosperm and angiosperm, follow this pattern with some variations where in the gametophytic phase is few to multi-celled. Point 3. Bryophytes and Teridophytes interestingly exhibit an intermediate condition, that is haplodiplontic. Both phases are multicellular, however, they differ in their dominant phase. A dominant independent photosynthetic thyroid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular saprophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern, given in figure 3.7c. Interestingly, while most algal genera are haplontic, some of them, such as Ectocorpus polycyphonia kelps, are haplodiplontic. Fucus alga is diplontic. Summary: plant kingdom include algae, bryophytes, pteridophytes, gymnosperm, and angiosperm. Algae are chlorophyll-bearing simple thyroid autotrophic and largely aquatic organism. Depending on the type of pigment possessed and the types of stored food, algae are classified into three classes, namely chlorophyce, phaeophyce, and rhodophyce. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may slow isogamy, anisogamy, or ogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structure. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axes bearing spirally arranged leaves. The main plant body of a bryophyte is gamete producing and is called a gametophyte. It bears the male sex organ called antheridia and female sex organ called archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophyte. In Teridophyte, the main plant body is Sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissue. The sporophyte bear sporangia, which produce spores. The spores germinate to form gametophyte, which require cool, damp places to grow. The gametophyte bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte, the gymnosperm, or the plant in which the ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked seeded plants. The gymnosperm produce microspores and megaspores, which are produced in mycosporangia and megasporangia, borne on the sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axes to form male and female cones, respectively. The pollen grains germinate, and pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote developed into embryo and the ovules into seeds. In angiosperm, the male sex organ stamen and female sex organ pistil are borne in a flower. Each stamen consists of a filament and anther. The anther produces pollen grains, male gametophyte after meiosis. The pistil consists of an ovary enclosing one to many ovules. With the ovule is the female gametophyte or embryo sac, which contain the egg cell. The pollen tube enters the embryo sac, where two male gametes are discharged. One male gamete fuses with egg cell, that is called syngamy, and the other fuses with the diploid secondary nucleus, that is triple fusion. This phenomena of two fusion is called double fertilization and is unique to angiosperm. The angiosperm are divided into two classes, the Dicotyledons and the Monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generation between gamete producing haploid gamete and spore producing diploid sporophyte. However, different plants group as well as individual may show different pattern of life cycle, that is haplontic, diplontic, or intermediate. Thank you friends for watching, for listening this audio. मिलते हैं नेक्स्ट वीडियो में, तब एंड मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय एंड please post your comment if you want any kind of improvements, and stay tuned with Biology Critic and enjoy the knowledge. Bye.